Hey everyone. Chapter 14 deals with the nervous tissue, in which we will understand the components of the nervous tissue. We're not going to go into details about the central nervous system and the peripheral nervous system and the nerves. Just for us to understand what are neurons and what are the glial cells that surround the neurons. So this is pretty much what we will be learning in this online lecture. Fairly easy stuff. We're not going to be dealing with action potential and electrical potential of the cells. All of that belongs to the physiology instead. So let's start with describing here the structural organization of the nervous system. We understand that the nervous system is comprised of central nervous system and peripheral nervous system. We're going to have about 3 or 4 lectures talking about the anatomy of the central nervous system. Also, we're going to have a couple of lectures dealing with the peripheral nervous system. Okay? So I'm going to skip through this slide because it just shows you here what is the central nervous system and what's the peripheral nervous system, which are not really our topic today. Again, it's just the anatomy of the nervous tissue. What makes a nervous tissue? These are the neurons and the glial cells, and they vary slightly between the central nervous system and the peripheral nervous system. Okay. So, function of the nervous system, it's either collecting information, sending information, responding to information, analyzing information. We're going to study that in more details when we get to the brain functions. We also know that there is what we call an afferent division, an efferent division. An afferent division is the division that will bring the information towards the central nervous system. An efferent division, essentially, it's motor, sometimes it's secretory, and that is the function that goes away from the central nervous system. If you're in doubt, so E, if you can remember it this way, E is with exit, so the efferent division will be the one that exits the central nervous system. Okay? All right. So the sensory nervous system, on the other hand, we're going to realize later on that we have somatic sensory and we have visceral sensory. Somatic sensory such as touch, temperature, pain. Visceral sensory such as blood pressure, oxygen concentration, pH. All of these things are considered, also blood pressure, are considered what we call visceral sensory division. We don't have to go into extreme details in that. In the physiology we will, but since we're dealing with just the nervous tissue from the anatomy perspective, we're not going to be spending too much time describing the different functions that we have in the nervous system. The motor, the same thing. So we have somatic motor and we have autonomic motor. The somatic motor, your skeletal muscles, your voluntary muscles, and your autonomic motor will be the movements of your stomach, movements of your intestine, secretion of glands, if we squeeze the smooth muscles surrounding them. All of that will be autonomic motor. Okay? So, now we're getting into the important stuff from our anatomy perspective. So the first thing we need to understand is that we have neurons. These are the nerve cells. These are the excitable cells. And then we have glial cells. They originally called it glia because they thought the only function for the glial cells was to glue the neurons together, so they called it glial cells. So the neurons, they are the excitable ones, but they don't divide. After you're born, you're done, you don't really divide. And whereas the glial cells, they are non-excitables, but they're highly dividing cells, and therefore they're good in repair in some tissues within our nervous system. We'll get into that quickly. And but they also, because they have high rate of multiplication, they have a higher risk of cancer development. So the cancer that we hear of, intracranial tumors, malignancies are generally glioma or glioblastoma, so things that are from glial origin. Okay, so the neurons, they are characterized by the following. They have high metabolic rate, and if you took physiology and if you remember from the cell biology, they have what we call the sodium-potassium pump, and the sodium-potassium pump is the reason really to maintain this polarity between the inside and the outside. Again, we don't have to go into extreme details in the physiology and the function of the neurons, but just for us to understand that they have high metabolic rate. Why is that? Because then when you have the brain being cut off oxygen, even for a very short little bit, that can result in death of the brain tissue, because it has extremely high metabolic rate and will not survive in the absence of oxygen for extended period of time. Okay? Has longevity. It lasts for all your life, except when in advanced age, when there will be neuropathy and there will be some atrophy of the neurons, or in the central nervous system, the person can start to suffer from, for example, Alzheimer. But otherwise, the neurons are designed to stay for as long as you're alive. Of course, now we are exceeding that time frame, so we're exceeding the life expectancy of the neurons, and that's why we start to see a lot of perhaps Alzheimer cases than we used to previously in previous decades. It's non-mitotic, that means it doesn't divide. Keep that in mind, because a damage to the body of the neuron, not the neuron branches. So if you cut the root of a tree, then the tree is going to die. You're not going to get another tree. If you cut a branch of the tree, then there is a chance to put that branch together, right? So keep that in mind, because when we get to the repair, we will talk about it. But for now, just realize those following characteristics. It has high metabolic rate, it's excitable, of course, it has extreme longevity, and it's non-mitotic. Okay? If we look at the neuron structures, we will realize that the neuron has the cell body, or what we call the soma, and then sticking out from it will be the dendrites, and then from the soma, then you will have the axon. Right at the axon junction with the cell body, you will have an area here called the axon hillock. This will be specifically important from the physiology perspective, because that is where the action potential is going to be generated, where the graded potential will be changed into action potential. We don't have to worry about the physiology, just focus on the anatomy. Here is the body, and here is the dendrites, or the dendrites sticking out of the cell body, and then we have the axon. The axon all the way towards the end, it can branch, and that will give you axon collateral, and near the end you will have smaller branches. We call them telodendria. So we have dendrites here, and we have telodendria all the way here, and then all the way at the very, very end, we will have a slight swelling. We call these guys synaptic knobs. Okay? So this is pretty much of the structure of a classical neuron. Of course, there are neurons that will look different. There are neurons that in which you will not be able to identify which one is axon and which one is dendrites, because they just have two sticks sticking from them, and you don't know which one is which one. We'll talk about those other neuronal structures, but these are the classical neuron structure anyways. Okay, so we have the soma, dendrites, axon. Axon can branch, gives you axon collaterals, and all the way at the end it can branch, also giving you, not always, branch, giving you what we call telodendria, and the telodendria will end up with the synaptic knobs. So in most of the neurons, they are myelinated, and we'll talk about the different ways to myelinate. That means they are insulated. In this case here, it's insulated by what we call a neurolemmocyte. Another name for the neurolemmocyte is the Schwann cells, and these are very important physiologically because they can prevent the leakage of the potassium ions and therefore the fast propagation of your action potential with minimal energy. Again, when it comes to physiology, you don't have to remember much since this is an anatomy lecture. Okay, so again, we have the inside the body, the cell body, we will have the nucleus, the nucleolus, of course, that's where the ribosomes are made. We have mitochondria. The rough endoplasmic reticulum is abundant, and it will show up under the microscope, and we call them the Nissl bodies. So the Nissl bodies is really rough endoplasmic reticulum of the neurons. Lipofuscin is a pigment that is present in neurons, especially as the neurons get older, and that gives the slightly yellowish coloration for the neurons. That's why in your anatomy book, neurons are always painted yellow, and that's because of the lipofuscin. We don't necessarily know what the lipofuscin is, and doesn't do anything, and as far as we know, and doesn't damage the neurons either. But now you know why we paint the neurons yellow in our book. The dendrites, generally, they are the place to receive the signal. So generally, again, I'm going to stress on generally. So the signal will come to the cell through the dendria and then to the cell body, and then it will be disseminated towards the axon, and from the axon, as you know, to the axon terminals and the telodendria and the synaptic knobs. Okay? So dendrites generally, again, generally, is the place where the signal really enters to the cell body. Okay, the axon. Some cells have a single axon, some cells have branching axons. We're gonna go into that later on. Some cells don't have axons at all, and the cells that don't have axons, we call them anaxonic. These are neurons without axons. And I'll show you some examples of unusual neurons later on. I told you about the axon hillock. That is the junction between the axon proper and the cell body. This is the funnel that I showed you earlier, and that will be important from a physiology aspect for the propagation or the generation of the action potential. From the axon, you will start to split the axon, and that will give you axon collaterals. All the way towards the end, it's going to branch into fine branches, and these will be called telodendria, and the ending of the telodendria will be slight swelling, expansion. At the very, very end of the telodendria, we call that synaptic knobs. Okay? So the structural classification of neurons, if I go here to this picture, you will realize there is one thing only sticking from that cell. You see here is the cell body, and only one thing is sticking out of it. And that thing will be, you can't call it dendrite, you can't call it axon, and so it's fairly hard to tell which one is axon and which one is dendrite, although in this illustration here we call them the dendrites just because of the flow of the information. Regardless, you can see here only one thing sticking out of the cell body, and therefore we call that unipolar. So one pole sticking out of the cell body, that's the unipolar. Bipolar, two things are sticking out from the cell body, and we see that, for example, in the eye, and also some olfactory neurons. There will be bipolar. And we have the multipolar, and the multipolar, look here how many things are sticking out from the cell body, right? Forget about the things that stick out later on. These are dendria or axon terminals or whatever. I'm talking about how many things are sticking out from the cell body itself. That will classify it to unipolar, and we'll see that a lot later on, especially when we get to the sensory neurons. We have tons of those, and this cell here will be right next to the spinal cord, whereas the neuron itself will be all the way towards your receiving the information from your skin or whatnot. We'll talk about that special type when we get to the sensory portion of our central nervous system. Most of the neurons are multipolar. Very few will be bipolar, and I gave you examples like in the retina, we have bipolar cells. Okay. So as I told you earlier, the functional classification will be either afferent, efferent. Afferent is receiving the information to the central nervous system, efferent sending out the information away from the central nervous system, and then you have interneurons, neurons that don't send the information out or in, but they just function between the neurons. We call them interneurons, and these are within the central nervous system. They don't leave the central nervous system, and they don't bring information towards the central nervous system, but these are the relay. You need to remember, however, that most of your neurons, over 90% of your neurons all over your body, are interneurons. They're not afferent, they're not efferent, but they're interneurons. Okay? So here is a very simple illustration of an efferent neuron. So this is a sensory neuron bringing it in, and efferent is the one that exiting the information out of your system, and the interneuron, this is too simplified drawing showing you it's the neuron that just passes the information from one group to another, and that of course is involved heavily in the memory and all the kind of things that you have, the little circuits that you have within your central nervous system. Okay, good. So we move from the neurons. Believe it or not, we now covered the neuronal structures, and now we are moving into the glia. The glia were the cells that glue the cells together, glue the neurons together. That's why it's called glials. And the glial cells have the ability to divide. They are non-excitable cells, although some of them can manipulate the existing synapses. For example, the astrocytes do. And but they are not excitable cells themselves. They don't form synapses with the neurons because they're not neurons. And we will see that we have different types. We have those glial cells inside the central nervous system, and we have glial cells outside the central nervous system, or in what we call the peripheral nervous system. So we're going to start with the central nervous system. We will see this cell here, and we call that astrocyte. And then we have a tiny little cell. We call that, because it's small, we call it micro, so it's microglia. And then we also have cells with multiple feet, and so it becomes your oligo, that's multiple, dendro, feet, cytes, so it's the cell with multiple feet. And we will talk about the function of each of them. And finally, we have the ependymal cells. So this is just a cartoon showing you those structures again: astrocytes, microglia, oligodendrocytes, and ependymal cells. So these are your glial cells of the central nervous system. We're going to talk about each of them independently. So the astrocytes, they have lots of functions. They help in functional blood-brain barrier, functionally, not physically. They don't participate physically in the blood-brain barrier. They regulate the tissue fluid composition surrounding the neurons inside the nervous system, central nervous system. They can help with the establishment of the synapses or inhibiting synapses. They can make scar tissue when there is a damage inside the central nervous system. The astrocytes are the one that giving you the scar tissue because you don't have fibroblasts there. They also assist in the neuronal development, and they will guide the neurons towards their terminal destination. So they're very important during development in guiding where the neurons are going. So keep that in mind. These are the functions of your astrocytes. And this is again the shape of the astrocyte and their relation with the neurons. The next cell we need to know is the ependymal cells, and the ependymal cells are only present wherever there is a cavity inside the brain. And yes, there are fluid filled cavities inside the brain. We're going to study that together, inside your central nervous system rather, and we're going to study that together. They're called ventricles. And those the big ones are called ventricles, and those ventricles will be lined with the ependymal cells. Okay, so you will only find the ependymal cells in the lining wherever you have the canals and wherever you have the ventricles inside your central nervous system. They have a function in the okay. So these were the functions of your ependymal cells. And again, as I said earlier, these they're also characterized by the fact that they have cilia, and the beating of the cilia can help in the circulation of your cerebrospinal fluid. And some research now is suggesting that the ependymal cells are your stem cells that are capable of making new neurons. This is still new data and it's still yet to be proven, but some people believe in the fact that these are the ependymal cells. So like I said, these cuboidal cells are lining wherever you have canals or wherever you have ventricles, and they have cilia, and so therefore they help in the reconditioning of your cerebrospinal fluid. Also, the beating of the cilia will help in the circulation of the cerebrospinal fluid. And finally, people think that the ependymal cells are some kind of stem cells. Okay, so now we get to the microglia. Microglial cells are your immune cells, so they are similar to macrophages outside. They are phagocytic cells. They wander around, they don't sit in one place, and they help in removing debris from dead cells or infection, if there is infection to that point. And so they're the smallest of them, and this is a picture here or a drawing of your microglia. And finally, the oligodendrocytes. Oligodendrocytes, they have a unique characteristics that their feet will become flat and they will start wrapping around neurons, not the whole cell, just the feet will run around it, and therefore providing the myelin sheath. Those cells and the myelin they produce, it's only present in the central nervous system and not the peripheral one, and they can be mistaken by your immune system to be an antigen, and therefore you start building antibodies against it. And we see that in multiple sclerosis when your own antibodies start to attack the oligodendrocytes and the myelin sheath that they are producing. But just note here that it's not the entire cell that is wrapping itself over and over and over around. You can see how many times it wraps itself around a neuron here, around an axon, but only the feet, okay? So these are our oligodendrocytes, and the function is establishment of the myelin sheath inside the central nervous system. We're good? Then we get to the glial cells of the peripheral nervous system. We have two types, the satellite cells and the neurolemmocytes. The satellite cells, they are present around the neurons outside the central nervous system, like in ganglia, dorsal ganglia and whatnot. And these cells will just recondition the fluid and help in the nutrition of those neuron cells, the bodies of the neurons outside the central nervous system, and we call them satellite cells. I'll show you a picture in just a short bit. The neurolemmocytes, another name for them is the Schwann cells, and these are the ones that produce the myelin sheath outside the central nervous system. So we have the neurolemmocytes for the myelin sheath in the peripheral nervous system, and we have the oligodendrocytes for the myelin sheath inside the central nervous system. Okay, so here's a picture here of the satellite cells around the neuron bodies in a ganglion, for example here in the dorsal root ganglion, and again its function, it's for the nutrition and the reconditioning the fluid surrounding the cell bodies. And whereas the neurolemmocytes, another name is the Schwann cells, they form the peripheral nervous system myelination around the peripheral nervous system. They're very important in the conduction, the nerve conduction. They're also very important in the repair of the neurons. They will assist in your repair, whereas the oligodendrocytes actually will prevent the repair. So therefore, when someone gets a nerve damage, it can be repaired, whereas when someone gets a central nervous system damage in the spinal cord and whatnot, we don't have Schwann cells there, so the damage is irreparable. Okay, so here is an example here how the Schwann cells or the neurolemmocytes wrap not only their feet like the oligodendrocytes, but the entire cell keeps wrapping itself over and over and over, ending up with what we call the myelin sheath. Between the myelin sheath and each other, so between the Schwann cells rather and each other, we're going to have nodes, and we call these nodes nodes of Ranvier. The guy who discovered these nodes, his name was Ranvier, and so these are the little intervals here between the neurolemmocytes, or between the Schwann cells. Sounds good? Okay, so that's just a quick comparison between the myelination inside the central nervous system by the oligodendrocytes and the myelination in the peripheral nervous system by the Schwann cells, or the neurolemmocytes. As I told you earlier, the Schwann cells have this unique ability to help in the axon regeneration whenever there is a damage in the cell, in the neuron. Then anything distal to the damage, you know what's distal and you know what's proximal, anything distal to that damage will start degrading, and then new neurolemmocytes or Schwann cells will start multiplying, and they form empty tube, and that empty tube will start to guide the neuron ending to where it's supposed to go. The axon is going to start moving on all the way here to fill that tube and reach the terminal area where it's supposed to go. So they're very, very important. The Schwann cells, very important in the nerve regeneration, because they have also high rate of multiplication. If you remember, we said that the neurons are amitotic, but the glial cells, they have high rate of mitosis, so it's not uncommon to see a tumor called schwannoma that's attached to a nerve, right? So just keep that in mind. When it comes to the nerves themselves, the nerve structure will have connective tissue surrounding the nerve, surrounding the nerve bundles, the neuron bundles, and surrounding the individual neuron. So we need to know them, just like we knew in the muscles, the epimysium, the perimysium, and the endomysium. Also here we're going to have the endoneurium. This is the connective tissue that surrounds the individual neuron fiber or neuron cell, axon. And then we have the perineurium that will surround the fascicles, just like we had with the muscles, and the epineurium. So it goes endo, peri, epi. And so the structure would look like that. So here we have a single cell, single axon. And by the way, the cell membrane in a neuron we sometimes refer to as the neurolemma. You remember the sarcolemma in the muscle? So here we call it neurolemma. Outside that we will have the endoneurium, and then you bundle few neurons together, neuron axons together, and that will be that bundle here will be your perineurium. Around the entire nerve you will have the epineurium. Sounds good? Good. So synapses are places, we're not going to go into extreme details because it's just anatomy. We have, it's the area where the signal will terminate, but the neurons are not touching each other. So keep that in mind. The two neurons are not going to touch each other, but instead you're going to send the signal, and the signal is going to travel the gap, and therefore the signal goes in one direction only. Because of that signal, it's going to divide the neuron into a neuron before the synapse and the neuron after the synapse. So we're going to have presynaptic neuron, and we have a postsynaptic neuron. So this is a presynaptic neuron. Physiology of it is not important. The neurotransmitter here, not important for you from an anatomy perspective. The neurotransmitter here is in vesicles. When the calcium comes in, after the action potential stimulates it, then the calcium will bind to a certain protein, and that protein will cause exocytosis of those vesicles. The exocytosis of the vesicles will result in the release of the neurotransmitter, and now it binds to a receptor and activates the cell afterwards. Whether this cell is a muscle, or whether a muscle fiber, or it's another neuron, like here in synapse, it doesn't really matter. You pass the signal from here to there. We're good? The space between them, it's called synaptic cleft, and sometimes the endoneurium will, or not sometimes, always the endoneurium will encapsulate this, so it will be continuous. The endoneurium will be continuous with the presynaptic and the postsynaptic. Therefore, they don't snap away from each other. You want them to be snuggling with each other. And sometimes we have adhesion here, molecules that will make sure that these two don't wander too far from each other. You don't want the, what we call the synaptic cliff, to be too far away from each other. Sounds good? This is what we call the chemical the chemical synapse. The electrical synapse doesn't have all that. It just holds between them. And if you remember from the first chapter, we covered those holes we call connexons or gap junction, and the gap junctions will just allow the signal or the electrical signal to pass from one end to another. This is a very rare type. It's not a common type. You see that only very, very early in development, and maybe in very rare places like the teeth, but they're very rare because they can allow the signal to go back and forth, whereas this one only allows it in a single direction. Okay, so this is a simple synapse, what we know of a presynaptic, a postsynaptic, and so here is a simplified drawing. The synapse is not always that easy. Sometimes a single neuron will receive about 10,000 or 20,000 neurons communicating with them. So it's a much, much complicated drawing if we are ever to draw it. But however, so we have, if the axon terminates with the body, which is the soma, we call that synapse axosomatic, because axon and soma, axosomatic. We have axodendritic, so it's between the axon and dendria. We have axoaxonic, so between the axon and another axon. So we have different ways how the neurons will communicate with each other. How they build the circuit, just like you're making an electrical circuit. There is the simple circuit is what we saw here. This is a very simple circuit. It goes in one direction. Sometimes the circuit will converge or diverge. So you start with 5 and you end up with 1. That's a converging a circuit. Diverging, you start with 1 ending up with 2000. That's a divergent circuit. You have something we call the reverberating circuit. If you look at it, you don't have to go into too much details because that's not physiology. You go from one end and then all the way towards the end you will have an axon collateral here sending the signal backward, and so this signal will stimulate that neuron again, so you keep stimulating over and over and over the neuron at different levels, maybe here at the axon collateral and maybe here after the cell already has given to a postsynaptic neuron. Then the postsynaptic neuron will fire back and creating to you this reverberating signal. There is also something called parallel after discharge. This here will be a very fast route because it's a single axon, but you can also send it through 3 different cells, and of course it takes longer time. And so by the time this stimulus will reach here, then very shortly after, milliseconds later, you will have another signal coming through here, another signal coming through here, and the third signal coming from here. We call that kind parallel after discharge. Okay? All right, so that was my presentation to you guys. I hope you find the information useful, so we can wrap up this chapter and focus on the bigger chapters ahead of us for the anatomy course. Have a great day.